Hello everyone. This is Devomani Ganguly from JIS Institute of Advanced Studies and Research, Kolkata. Welcome to you all to the fifth lecture of the annual Distinguished Lecture Series 2021, organized by our Center for Health Science and Technology throughout the month of August. This lecture series is organized as a part of the celebration of the second Foundation Day of our institute. I hope you have enjoyed the previous lectures and eagerly waiting for the remaining talks. I again request you to browse our brochure to know more about our institute, JISIASR, and the courses we are running. We have MSc in Medical Biotechnology and Bioinformatics, Advanced Diploma in Bioinformatics, and PhD programs. Detailed information regarding admission can be found in our website. All contact information is given there and also visible on screen. Email us or call us for further information. It gives me immense pleasure to grace all of your presence today in the fifth lecture of the series by Professor Dibanko Chatterjee. Thank you so much, sir, for accepting our invitation and being with us today. Live streaming of this talk is available on our YouTube channel, CHEST, JISIASR. Please write your questions to the chat section of Zoom or comment section of YouTube. May I now request Dr. Kamakshi Surekha to introduce our today's speaker, Professor Chatterjee. Thank you so much. Over to you, Dr. Surekha.
Thank you, Dr. Devovani. It is actually an honor for me to welcome and introduce Professor Dibanko Chatterjee, a name that actually does not require any introduction. He is well known for his pioneering research on bacterial transcription. But for the record, I have to mention these things. He did his master's from Jadavpur University and PhD from IISc Bangalore. After doing his postdoctoral studies from Albert Einstein College of Medicine and Stony Brook University, he joined Molecular Biophysics unit of IISc as faculty, where he did most of his remarkable works. He is a recipient of Shanti Swarup Bhatnagar Prize and is an elected fellow of all the major Indian science academies. The government of India awarded him with the Padma Shri in 2016 for his contributions to science and engineering. I am a big fan of his works from the days of my PhD and following his phenomenal research on exploring bacterial transcription mechanism and genome expression in E. coli and Mycobacterium tuberculosis. His work on omega factor needs a special mention. We are so fortunate to have him with us, and today he will be talking about transcription machinery of E. coli and the effect of silent mutation. So without any further delay, I am welcoming Professor Chatterjee. Sir, the stage is all yours.
Thank you very much, Kamakshi. I will share the screen now.
Sure, sir.
Right. Is it coming full screen?
Yes, sir.
Okay. Thank you. And so unfortunate that I am giving the first talk of your second Foundation Day, my first visit online, which I don't like at all. I'd love to see the place, love to see the department, library, and students meet faculty, but someday we'll do that. When COVID, we are free to do so. We are now having problem with you, all of you know that disease is killing all of us. It is a pleasure to give this talk, and Kamakshi told us about and Kamakshi and Devovani told us about the new institute. I'm sure you are all enjoying your studies. I don't know how you handled it. I was discussing with them how it's difficult to give online talk because we do not interact with the students. It is more difficult for them to conduct an exam, set up a paper and give a class. I'm sure all of the teachers who are present here today and the students will understand this. but we have to utilize the best possible way we can do that in the present day situation. What I'm going to do, I want to talk, is a very well-known subject, transcription. Now it is, everybody knows about transcription. The students of biology, particularly, and undergraduate students. I will try to put some of our effort on transcription, what we are doing in bacterial transcription, and exactly why you are doing this work. We have been doing this for a very long time, so it is indeed a pleasure for me to talk on this subject. If you want to have any question, you can always raise your hand and stop me and ask question, or you can put a question in your queue. Then after the talk, somebody will organize the questions and ask me. Or you can send me my email, which is easily available with Kamakshi and others. So transcription is a complex phenomena. It is basically DNA to RNA to protein is the central dogma of molecular biology, which was proposed by Francis Crick in 20th century, one of the best known biologists, probably the best biologist of the 20th century. And now there is an institute in his name in England, Crick's Institute. He proposed central dogma where DNA makes RNA, RNA makes protein, and protein is the functional unit. So the first step, DNA to RNA, is transcription, and RNA to protein is translation. And why is it called central dogma? It is very interesting. When people were analyzing after 50 years of central dogma proposal last year, they said it is so interestingly coined the word dogma. The dogma of the whole thing is that once the proteins are made, they will not go back to the DNA again. It is locked in the protein conformation. It will never go back. RNA to DNA is possible, but protein to DNA is not reversible. So that is the dogma of the whole concept of molecular biology, central dogma. But we will not go into the protein. We will look at the RNA synthesis and what is the mechanism of doing so. Every living organism follow this pattern with different degree of complexity. In case of a virus, it made by one subunit, one particular protein to make the RNA from DNA. In the case of bacteria, it is more than one subunit. I'll show you very soon. But in case of eukaryotes like us, or yeast, or Drosophila, you name it, there are 3 different kinds of polymerase doing 3 different functions. So the complexity increases, but the basic mechanism remains same. And I'll try to point this out as I proceed in my lecture. I hope I am audible and clear, and if you have any problem, Kamakshi, tell me. It is your duty to tell me that whether it is audible or not. I'm doing
so everything is fine. You are perfectly audible.
Okay, but stop me anytime you think that I'm going too fast or anything. Okay?
Sure, sir.
Okay. In the next slide, I'll show you that my slide is not moving now. 1 minute. Yeah. These are the subunits in the case of E. coli or any bacteria. There I have written in the top, there are 2 alpha subunits. I put alpha 1, alpha 2, beta, beta prime, and a small subunit omega. I'll talk mostly on this particular subunit. But these are the major subunits which come all together. In the case of bacteria, these are the subunits. There are 2 alpha subunits. They are same gene product, but they have little different function. In the case of archaea, there are variations of beta, beta prime gene, but their name is like that. In the case of omega, RPOK mimics the function of omega, but still is only 1 protein. In the case of eukaryotes, there are 3 proteins, does ribosomal RNA, messenger RNA, and transfer RNA synthesis differentially. And these are omega counterparts there, which is RPD6. So, this complexity, as I mentioned in the beginning, complexity goes up, but function remains same. So people believe that if you study one enzyme very particularly in great detail, you will know the function of how RNA polymerase works. This is a crystal structure, 4.2 angstrom resolution, little old, but now it is little better. The structure is known. And alpha directs enzyme to the promoter, sigma subunit, which directs enzyme to the promoter. Whole enzyme is called coenzyme, which without sigma subunit. The coenzyme doesn't do any promoter-dependent function. Now, you can ask me, undergraduate who is sitting here in my lecture, that what is a promoter? Promoter is nothing but which promotes DNA synthesis, gene synthesis, DNA to RNA polymerase. DNA to RNA by RNA polymerase. The promoter is selection of the promoter sequence. RNA polymerase does the promoter sequence selection. There are certain base pairs which are very classically oriented for this synthesis, and they make RNA from a particular sequence. You understand the problem that if the synthesis is done from anywhere in the DNA chain, you will get a nonsensical message. Stop codons will come. No protein will be functional. So it is exactly the location of the RNA synthesis, where from it should start, we call it +1 site, is directed by upstream or some sequences up, away from towards 5' end from the +1 site, which are called promoter site. It is almost like a promoter or house building construction. We have promoter for flat houses. It's basically the same, who promotes the synthesis of RNA from DNA chain. So sigma directs the RNA polymerase. We have worked extensively on this subunit, and there are different kinds of sigma for different gene sequences. It goes to the enzyme. Beta prime is involved in catalysis, chain initiation, and elongation. Then beta involved in chain initiation and elongation again. Further, alpha is required for assembly. Alpha is take the whole RNA polymerase assembly takes place on alpha. And this is the omega subunit, which function remains very difficult to understand. What was it? Why the confusion of omega? In fact, I must tell you this. Nobody believed that omega is a subunit. When it was isolated, people found the RNA polymerase preparation comes with omega. People thought it is a contamination. It comes in the, many times in your laboratory, you make a protein and some other protein comes with it. People thought it's a contamination because when you take away this alpha, beta prime, all the sigma separately, purify them separately and mix them together, you find the RNA synthesis happening from a particular promoter. So, the question is that this is called in vitro reconstitution. So what is the problem? If the in vitro reconstitution doesn't require, enzyme doesn't require omega, then what is the function of in vivo? That question comes. So the question of its function remains tightly bound impurity, 1969, long back. None of you were born at that time. RNA polymerase lacking omega retains. Uncompromised transcriptional activity is very important. Reconstitution of RNA polymerase devoid of omega show no alteration in transcription. Insertional inactivation or deletion RPOZ do not affect survival of the cell. So if RPOZ gene makes omega, if I do insertion mutation in the RPOZ gene, it will be altered, and still you see the transcription going on. But still, Richard Burgess from Wisconsin, when I was doing this work, he asked the question, but then why every preparation has omega? It must be having some function. But the validation came from Burgess's work. Burgess first showed that omega binds tightly to the beta prime subunit, cross-links. Omega acts as a transcriptional activator when omega is a subunit associated with RNA polymerase. So omega requires to activate the transcription, so it cannot happen without, if it is not a functional part of the enzyme. And we showed several years back, in any organism, that if you remove omega from RNA polymerase is inactive. We showed it is inactive. However, it remains active. People miss that point that whenever you remove omega, GROEL comes with RNA polymerase. So GROEL is an unavoidable part of RNA polymerase if omega is removed. So therefore, question, all of you know, perhaps who do not know, let me tell you, GROEL is a chaperone, is a global chaperone, which helps the RNA polymerase to fold. So people thought at that time, including us, that GROEL, therefore, omega must be having a similar function like GROEL. So here comes the little biology I'll put it to the students. I'll start with this and then I'll go back to my experimental work. That Problem is, you understand how to, it's very easy to address this kind of problem in a bacteria nowadays because genetics is so strong. That if you find some protein is required or not, if you want to show that, the protein should remain associated with the enzyme to have the function. If the protein you remove, the enzyme is functional.
Excuse me, sir. I guess.
Okay now?
Yes, sir.
So shall I repeat something?
We just missed few seconds. So.
Okay. So what I'm telling, this is a strategy we have to follow. We followed, which is a pure bacterial genetics. Where what we do, that what is my point? My point is to show that omega is a contaminant or it is a subunit. Like this question is very important and valid question. To prove the problem becomes, if we remove omega and do this work, we find that some other protein role comes. So how do I pinpoint the function? So we developed a strategy called toxic mutant strategy or called dominant negative strategy, where we did a mutation in RPOZ gene, which makes omega. You try to follow this point. We make a mutation in RPOZ, which is a created toxic mutant. At the same time, it also tightly bound with the rest of the enzyme. We make a RPOZ insertional mutation, random mutagenesis, and try to score with the several screens, try to score a mutation, which will create is functionally incompetent, yet remain bound with the enzyme. If it remains bound with the enzyme, rest of the enzyme, and become functionally incompetent, then you score what function is incompetent. Then you know that therefore RPOZ must be having some role in that function. So if one isolates a toxic mutant of RPOZ, that is the mutant I'm telling dominant negative, one way in which it is toxic would be that the mutant may remain competent with wild type omega to gain entry into RNAP and core. Such a mutant RPOZ protein now proves that RPOZ is a bona fide essential subunit called dominant lethal. Such a system also allows us to examine situation where grow aid is prevented from function. So in the background of the wild type omega. So there is we are creating omega by creating a toxic mutant. This point is very important in my whole talk. That mutant now competed the wild type omega, but it remains strongly bound with the RNA polymerase. And you look at the function, then we know what is the function. So it's a typical genetic approach. You put in a reverse argument, you try to put the function of a particular protein. So you go ahead with that. Now this is one point of the toxic mutant. Another point I would like to mention, the omega secondary structure. The omega as such has a little disordered structure. I don't have to go for the analysis, how do you do that analysis. However, when it comes to the enzyme, some helicity and certain function comes in. So the disordered proteins sometimes are very important. We call it, I will name it as internally disordered protein later. But these proteins have a disordered ordered conformation changes happening and binding to the core enzyme. So the structural conformation of the omega, such as 91 amino acid protein, such a small amino acid protein, is structured and its helicity has an important role to play its function and being bound. If the protein is very strongly bound, the structure alteration changes, and it is plastic in a way in original sequence. And if you do this analysis, you will find the same situation with the protein. So this is a very important point. Please remember, I'll show many results of that. In the meantime, I have also carried out some work to find out effect and presence and absence of RPOZ gene. If we ask the question, we are in one hand, we ask the question of isolating toxic mutant. On the other hand, we are also trying to see if you remove RPOZ gene, we want to see what happens to the enzyme. What are the function happen? What are the major thing happen in E. coli? See, in order to do so, I did one of my collaborators from Japan, Akira Ishiyama and Yamamoto, we did extensive search in 2017-18 and look at a chip sequence. These are basically E. coli whole genome is available. So we can find out what are the genes get altered, or which are differentially regulated in the absence and presence of omega. So how, come to long story to short, I don't discuss this work, this is involved work. But what we did, we want to see the presence and absence of omega, how they control the basically the differential gene expression in the back, right? E. coli in the background. So defined distribution of omega wild type and RPOZ defective RNA polymerase. We show that RNA polymerase, and we located various genes which are, I don't know further details on this. Various genes which are differentially regulated. So the number was mind-boggling for us. That means we show that it must be having important function. That so many genes are getting impaired in function or upregulated by RPOZ deletion. And what is more important that we show, there are some virus-induced genes that when virus infect, E. coli bacteria is infected with the E. coli bacterial virus, we find that RPOZ play a very important role. It has a tremendous ramification in today's situation, that why omega is influencing the virus-induced induction or infectivity by the virus and the RPOZ modulation. So we look at the microarray. We did a phenotypic microarray. We have this facility in our laboratory, where we look at the functional growth of omega, absence and presence of omega in presence of different antibiotics, and we showed how differential gene expression and different antibiotic presence and absence play important role. So all these last 3-4 slides is a very important work. I'm not talking about this work. What I'm going to present to you today, the function of omega. In order to do so, I said toxic mutant screening, that we created a mutant, which is very important. If we create a mutant in the omega in such a way, there is a toxic mutant, which remain attached to the enzyme very tightly compared to the wild type. And with the impairment of certain function, it will not follow up to accommodate grow yield. And this function will tell me what is the original function of omega in a concocted, in a reverse way. It's a genetic approach. I also showed the second important point, which students should remember, that circular dichroism studies or some other structural studies are necessary to find out what is happening with the enzyme upon binding, free omega binding to RNA polymerase, because there is this conformation change happening in omega and structure to structure. So enzyme has a particular structure. In the free form, it is a different structure. Okay. With this background, I'll come to the next thing which is happening in this. So when we did the toxic mutant screening, we worked on this system extensively, and we found out several mutants, single base mutation happening. We named them omega 2, omega 6, omega 8, omega 9, all that. One particular mutation we worked extensively because it was coming all the time. N60d mutation is the one delta change, one Dalton change takes place. Then so to code this is very difficult, but we did it. N60d mutation happens. Omega 6 mutation we call it. Other mutation we don't look at it right now. Just keep this in mind. So this is a lethal mutant. Please remember, write down in your notebook if necessary. This is a lethal mutant. This lethal mutant is creating toxic mutant, but there is an amino acid change is happening here. In addition, we are also having mutation, which are called silent mutation. I show here silent mutation. What is a silent mutation? All the students will know there is a degenerate codon. For 20 amino acids, therefore 64 possible combinations are there, triplets are there. So some triplets are very heavily used, but there are degeneracies. So I can have nobody will disagree with me, all the students. I can have a mutation in the DNA sequence, subsequently mutation in the RNA sequence, but no change in the protein because the two mutated triplet code for the same amino acid. That is called mutation, but it's a silent mutation. It's a beautiful name. So we also found some silent mutation at omega 9. Of course, is a toxic mutant like omega 6, which has a bona fide mutation into N60D. But this silent mutation also is created. And I'll talk about it, why you worked on this later. So we asked the question, and we are worried about it for a few years when we asked the question, but it's a very difficult question to answer. So we concentrated on N60d mutation, which is a toxic mutant and is a single amino acid change. So it is easy to work for that. We try to decipher the system. Omega 9 is a silent mutation. We only asked a question. We had no clue on that time. What is it that makes it a dominant lethal mutation? You understand the question? Omega 9 is a silent mutation. So keep your mind ready for this story. That I have a wild type protein, I have amino acid omega, where I have a change in the omega 6, where it is a toxic mutant with one base change, but making a different amino acid, N60th position, N becomes a D. So it is having a following toxic mutant, titer binding, blah, 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 some function. But that we answered, that question we answered, subsequent slides. But we kept a question in our mind, we had no clue to answer it. That omega 9 is a silent mutation. Omega remains same, like there is no change in the amino acid. Only the base sequence of the DNA and RNA change. So we asked the question, if omega protein remains same, why is that it makes a dominant lethal mutation? We thought it's a very important question. This is a characterization of N60d. I'm going to talk about N60d more for the time being before I jump to silent mutation. It shows that it is sluggish growth pattern, and we induce it in this slide, GL shows the RNA level of RNA. It is a sluggish growth pattern, and but still we can isolate the RNA and the protein from that. So total RNA profile. And we found that if there is a mutation N60d, concomitantly we try to find out a suppressor for that mutation. So on the background of that particular mutation, we created a suppressor of omega 6, and we looked for a suppressor. So we got a wild type mutation. Wild type, we have a N60d single amino acid mutation, and we got a suppressor mutation. And strategy following the same way. We finally got all the suppression and everything. When we looked at the suppressor, we find the suppressor, you expect it. It is nothing new. We expect that this omega binds to beta prime, as Dipankar just showed. It binds to beta prime with the N60d mutation, but the suppressor mutation binds to beta prime. The mutation comes in position at Y, a tyrosine, just next to NADFDGD. That is the catalytic site of the classical catalytic center of RNA polymerase. And omega suppressor, N60d suppressor comes. Why I'm showing this slide? This tells you, forget about the technique of creation of suppressor. This particular result tells me that omega is very important. I am getting a toxic mutant N60d mutation, but this suppressor is mapping at tyrosine, which is connected in the beta prime catalytic center. And therefore, omega must be having a functional significance, because the catalytic center is the suppressor mapping. So these are basic genetics. So we went ahead further. So we isolated omega from suppressor. We isolated in subunits. We reconstituted the enzyme and look at it functional studies. When we look at the functional studies, we find RNA synthesis is going on wonderfully. However, in the presence of the classical inhibitor rifampicin, it is inhibited. So it is behaving like a proper RNA polymerase, which is reconstituted enzyme with the suppressor and N60d mutation, which is not happening in the presence. See, you can see that if I have omega 6 RNA polymerase, if I have suppressor only, then only the transcription will happen. But otherwise it will not, so on and so forth. So I hope you are following my talk. Am I okay, Kamakshi? Are you hearing me? Hello?
Yes, sir. Yes, sir.
No problem, right?
No.
Anytime you become inaudible, you let me know.
Sure.
Now, so continue to write. we have done several transcription assays and showed. So to cut the long story short, in gist I'll put it, that if I have omega-6 mutation, transcription is impaired. But if I have a suppressor which forms in beta prime subunit where omega catalytic center is, then together they make it active. There is a beauty of suppressor genetics that a double negative makes a positive. One mutant is not working. But if I suppress, generate another mutant which is acting as a suppressor on the original one, then two together will be functional. So he did this work and several years back when he published it. But what is important that I showed you that structure, that any mutant we are finding, they are having omega-6 and silent mutant N60d mutation, 82% alpha helicity. Or in other words, we are finding that this is alpha helical in nature, omega-6 82%, whereas originally is not so alpha helical. So lethality is connected with the structure element of the omega, which is a very interesting finding. We modeled, this is the Y, the suppressor tyrosine, where the near the catalytic site, and this is the active site of the enzyme. We modeled in the whole omega where is the mutant and where is the catalytic, where is the suppressor site is mapping. And we looked at it, and it's a small video. I think it works. If the video is working, I do not know. Yeah, so you can see that you can look at the active site, you can see how open the site is, and you can look at, you can pinpoint the active site and you see that how open the site is, and this is how the whole omega suppressor complex comes is right in the catalytic center, and this is the omega catalytic metal. So that's how the whole story is on the case of transcription. And comparative binding affinity, obviously I told you that omega binds RNA polymerase, but we talk weakly because if I want to remove it, I can remove it easily and GROEL comes. But if I have omega-6 mutant, it doesn't, it binds very tightly with alpha helical structure. So if the structure is alpha helical, it goes and binds RNA polymerase very tightly, core enzyme, whereas native omega is plastic, doesn't bind it, acquires structure after getting incorporated, but is getting weaker binding. So omega-6 is tightly bind, we found the binding constant, etc. We did 3 different physical techniques. I just want to show you how we did the technique. We did a surface chemistry Langmuir-Blodgett technique to find out the binding constant. We did surface plasmon resonance to find out the binding constant. We did calorimetry to find out the binding constant, and everywhere we got the same result, that a differential affinity of the mutant omega and omega-6. So N60d mutation told us everything about the binding nature of omega with RNA polymerase. Cite one single particular mutation changing the structure, and as the structure is changing, that RNA polymerase activity also changing. Lethality is changing, which is showing so much consequences of differential gene expression in so many genes I showed you at the beginning of my talk. So, and also the recognition of the RNA polymerase with different omega, either omega-6 or normal omega, wild type omega. The sigma recognition also alters, as a result of which the promoter selectivity alters. So it's the whole mind-boggling ball game. We opens up such a beautiful experimental results that how omega can control the transcription. With one toxic mutant scheme is so easy to isolate. All the students, please remember, if you are doing bacterial work, please do bacterial genetics. Take the help of bacterial genetics other than other experimental work. But that is easy to manipulate those experiments. You have to think clearly. You can create suppressors, and bacterial genetics will tell you. One dominant negative mutant will tell you a lot about functionality, and creation can be by random mutagenesis. Now the techniques are all available. Okay, so we so on and so forth. We did all the binding constants of plasticity. So interaction between omega-6 is defined highly by enthalpy and small by unfavorable negative entropy terms. The plasticity of the interacting domain from omega by omega-6 are compromised, which may be responsible for the entropic cost. So we did thermodynamics and handled the whole system well. However, this is going as an expected result. I told you this is going expected, what we wanted to do and what we thought of doing. A toxic mutant we created, we showed the structural alteration by circular degradation. We did the biophysical characterization of the binding. We did this genetic differences, differential gene expression. We looked at it, so on and so forth. So we characterized it very well. We did not answer one question: what is about silent mutation? N60d is not a silent mutation. It's the mutation which happens, change of amino acid happening. Therefore, the structure is altering. What is happening with the silent mutation? Now come the silent mutation stop. See, you can see that there is no mutation. There are point mutation, can be silent, can be nonsense, can be missense. And silent mutation, I am the same lysine, but I am having TTC convert to TTT. So here the 64 codon, if you look at the amino acid sequence, you will see many of these base sequences all the amino acids are degenerate arginines, like histidine, proline, leucine, most all of them. And as a result, the glycine itself has 4 codes. So if I can change one code to other, I can have the same protein, but still the functional implication is enormous. So, and one more important point, there is a codon usage bias. Certain triplets are very much favored than in case of other one. Therefore, like in case of arginine, AGA code is 22%, AGG is 23%, or CGU is 9%. So, you have differential expression, differential codon usage. So, you want to study this, you have to follow the codon usage pattern also. Here comes the very important point, that normal omega, I try to follow this point. Normal omega is unstructured, I told you. CD pattern tells us. Omega-6 N60d mutation showed CD like this. But when I make omega-9, which is a silent mutation, no change in amino acid sequence, I get CD pattern alters. I just did not believe this result because it's the same protein like this, right? No base change. Sorry, base change is happening. No amino acid change. So amino acid of the protein, 2 proteins are same, omega-9 and omega. But this is non-structural, this is alpha helical. When we purify the protein, omega and omega-9 look same. In GL you can see. But omega-9 has a 43% helical content, whereas omega as such has 16%. Omega-6 in N60d mutation has also very high helical content. So we know that single amino acid change and therefore protein is different, its functionality is different, and etc. etc. But how can silent mutation have same amino acid sequence, only one base change at the DNA level can give this phenotype? People don't believe me because this goes against Anfinsen hypothesis, Nobel Prize winning work. It doesn't go against it, but initially it appears. I'll explain to that to you. We did mass spectrometric analysis, see silent mutant and normal one at the same distribution. We know that they are same. So the question which comes repeatedly, both the proteins have the same amino acid sequence, then how are they taking differential secondary structure? That is a question which bothers us. So we looked at the codon usage index in the case of alanine, codon GCG 0.36%, and this alanine mutation is taking place. GCC is 0.27%, GCT is 0.16%, and omega-9 and omega, we did a bacterial growth and everything showed our observation is right, that omega-9 is not helping the RNA polymerase to function properly. So we asked the question, despite having the same amino acid sequence, how can they show different phenotype? Protein sequence is same. I'm repeatedly telling this point that I am having changes happening here, but these changes giving rise to same amino acid, but yet the protein structure is altered. and functionally it is different. What did Anfinsen do? The experiment, Nobel Prize winning work. Anfinsen showed that if you take native ribonuclease, there are 8 cysteine, 4 sulfide double bonds. If you denature them, they all are 1, 2, 3, 4, 5, 6, 7, 8. They all open up. Now, if you slowly cool back, they become reconstituted to enzyme. You can follow by activity. But if you do rapid cooling, freeze it in ice bucket, you will get a 1% of the conformation, 1% of the activity called Campbell conformation. But that protein now, if you add little bit of markup ethanol or incubate at 37 degrees for a little bit of time, they go back their activity. Or in other words, the protein opens up, no activity, but they fold back exactly at the same way as the native one and allow them time. Or if you give a reducing agent to manipulate so that they can come back to their original sequence properly, right sulfide CC bonds are formed. So this gave him clue, 2 very important conclusions. First is the amino acid sequence tells you the protein folding pattern. Second one is that native sequence is the low energy state, the most stable state of any protein. It will try to go back there all the time. Folding will always try to go to the native state conformation. There are certain gaps, maybe you can push it to go in between, which are transient states, which are not permanent state. So we are not going against Anfinsen. To prove that point, what we did a simple experiment. We took omega-9 and we refolded it back, see the profile, we look at it after 10 years after refolding. So we get this is the omega-6, which I told you has a high half helical. It will never go back to native because it is a mutated protein, non-functional toxic mutant. Omega-9 is a toxic mutant also. So it is giving a alpha helical pattern, less alpha helical, but 43%, but still alpha helical. And this is a normal omega. But this protein now, if you take, and if you allow it to refold, or if you take this protein before and denature it again, like Anfinsen experiment, I denature it and refold it again, it will go back to the native structure. That made the point that in this structure, what we are getting is we are getting a structure which is toxic because it's a transient state, but given an opportunity and chance, it will go back to the native structure again. So these are the different helical content. Omega-6 is 82%, omega-9 is 33%, and helical content after refolding, it is 17%. So this alteration is happening by refolding helical content, by denaturation studies again. We thought we are doing something very weird, but we found no, it is not true. In case of internally registered proteins, several publications started coming in middle of 2000. Within last 10 years, so they showed the physical origin of codon positioning of the codon position and its occurrence, and the rarity of the codon position and the codon usage leads to the protein folding pattern. So with the support of these publications, literature, what we did, we try to see whether the position, and we made a mistake. If the position 81st where the mutation is happening and giving rise to silent mutation, then we wanted to see next position, the value, whether it is creating the same thing. But unfortunately, we found 82nd position, we do not create that. So it is not that position is important. It is the amino acid type, codon usage bias, and the beginning of what are the positions. It is important. We have to do systematically all the amino acid changes we have to do. It is 91 amino acids, so it is easy to do. So what we did, silent mutation, then we found in the literature, there are several evidence which shows that there is a protein solubility, mRNA folding, co-translational folding, protein production level, translational speed, transcription and translation regulatory elements. all can be controlled by silent mutations. There are several evidences are coming. It's a very mind-boggling. Several important investigations are coming. None is going against Anfinsen hypothesis. It is telling the silent mutation probably creating some proteins in a transient state, which goes to functional form, given a proper time and energy. So, if this is the RNA from which the amino acids are in ribosome is moving and translation process is going on, then there is a frequently used codons and less frequently used codons are there. So there is a slow reaction, fast moving, the polymerase will move continuously, and there is a slow and fast movement. And this movement will continue to be very important from frequently used codon to rarely used codon. So there is a eukaryotic RNA, so it's a 5' cap and there's a 3' poly A tail, and this folding RNA polymerase takes place. So how RNA polymerase is moving or ribosome is moving depends upon the nature of the codon and rate of movement, and the rate of folding will be guided. Therefore, if I have a DNA-based change, RNA-based change, rate of folding and the position will guide this folding of protein. So the protein will be folded little wrong way. But if you take the protein and denature it, it will go back to its native conformation again. But the rate of folding is entirely dependent upon the position where they are making the silent mutation and the rate of movement of the ribosome and RNA polymerase. So frequency of the code and position of the code both guide this. So put this point, there is a hare and tortoise race, you know that, how the RNA polymerase is moving, how they move. Some are waiting for some time, some are moving fast and waiting. And then this basically tells you how the protein folding is guided by the rate of ribosome movement and rate of RNA polymerase movement. So you look at the codon usage bias, arginine and isoleucine, we see there is a lot of difference in codon usage bias. So we did our very critical last experiment. So we took the strategy. The mutant frequency code 2 is rare codon. And this mutation should be generated at different position and from different amino acids. And arginine and isoleucine were selected, check for the protein lethality and change in protein structure. With our assays, all the assays are available in the laboratory. It was very easy to do that for us. So we generation of silent mutants, screen for structural changes, CD analysis, partial proteolysis, and look at the activity as in vitro transcription as a lethality. And we grouped it. Why do we group it? There's a group of mutation which created helical structure, lethal. Group of these are native, which is non-helical, and there are a group of mutation which doesn't root, which are non-lethal, and they have no plasticity, no structural alteration. So you know what are the position it is happening from this, but analyzing this. I will not ask you to go through all these slides, but this basically tells you 3 groups of mutation. One which behaves like a native one. One is a helical structure, and their functionality, their lethality exactly follow hand in hand. Then we did, my student did a wonderful experiment. What she did, she took and charged the system where we have a codon bias codon, which is altered codon, silent mutant codon. We charge the system, we call it codon plus strain, with a tRNA with a normal triplet. tRNAs that are helping the ribosome to making the amino acylation and translation of the first for the amino bond, the peptide bond as the translation goes on. So if I use codon plus strain, then my I have in the growing protein chain in the 50th subunit of ribosome, I have a amino acid tRNA containing silent mutant tRNA, but I have a codon plus strain. I put a lot of amino acids containing with the normal codon strain. So codon plus it is. So the codon which is coming in the tRNA, which is binding to the messenger RNA on the ribosome, that will have a normal counterpart with the in excess codon plus strain. And we find that it goes back to the native, it goes to the wild type. So even if I have omega 7, which is Green I can show is helical, lethal. If I use omega-7 codon plus strength, a lot of tRNA is coming from the normal counterpart. Then it goes back to the original form. So indicating, therefore, that our hypothesis was right. We did the colony formation assay to rescue the phenotype assay, which I will not bore you to death. And the final experiment, what we did, we reconstituted the RNA polymerase from different subunits and different position, and we dialyzed the protein, isolated the protein, and look at the transcriptional activity. And we isolated individual subunit. We found the suppressor map on the same position, catalytic position. So the functionality of omega remains same. So we found out the suppressor activity, and we found they are deformation sensitive and transcription goes on as expected. So I will not explain this slide. This is a simple result. Basically, the detailed experiment, which is single round transcription assay, told us that RNA polymerase is functional. And we also did proteolysis to show that omega and omega-7, they are same protein because there is a silent mutated protein, but their rate of hydrolysis is different. You can see here the difference in the rate of hydrolysis. So our hypothesis is right, which can go back to omega conformation once you go through a denaturation cycle, and you can get it functional by putting lot of codon plus tRNA. So they are not only differential, they are differential conformation between them, omega and omega-7, the same amino acid sequence. They go back to omega, everybody goes back to omega for many hypotheses, unfinished hypothesis. But the differential cleavage pattern tells us that conformation is different, which is expected, which we prove in several ways. So summary, I tell you, I'm finishing my talk, that internally disordered protein omega we consider is one internally disordered protein. They frequency codon interrupted by rare codons, and these codons, ribosomes, their movement is dependent upon the local translation kinetics, which is very much codon position dependent. And we get structured proteins and we get unstructured proteins. So the movement of ribosome and RNA polymerase on transcription and translation get impaired by the single base substitution. However, it is not the case when you have a excess of tRNA present from a codon plus string. Movement alters. So the rate of movement gives rise to altered protein conformation. However, if you allow them to further equilibrate within the initiation cycle, they'll go back to the wild type conformation again, which will be functional. So that is the story I wanted to tell you about the role of silent mutation. I'll end there, and this is the work done by two students, Unnati Patel and Sudhanshu Gautam, and one former student, Paramita Sarkar from Calcutta, and later Devaprita Bhaumik, a postdoc from Jadavpur University. Thank you very much.
Thank you, sir. I guess everybody is getting ready with their questions to write. But before
Was
that,
it clear?
I have few.
Was it clear, Kamakshi?
Sorry. Yes.
I was
totally
audible.
clear, sir.
I was audible, right?
Yes, fully.
Okay.
So let everyone get some time. So I have few questions to. So this is like phenomenal work. It's the whole protein folding during the translation. How important the timing is. It's really a phenomenal work, I must say. So so the GrowEL thing that came to my mind. So was it happening? Like helping the omega factor to fold correctly, or how come?
GrowEL helps it, but omega has a function like GrowEL. So if you remove GrowEL, omega actually doing the job of RNA polymerase folding. If you remove omega, then GrowEL comes and do the job. If you remove GrowEL and omega, the RNA polymerase is inactive. That will show.
Yes.
So omega is a chaperone. He's doing his function because he's self plastic protein. That's
okay.
But when you make a silent mutation, not that function is happening.
Right. So that function is impaired. And so that's why the total structure and plasticity of the protein is so much important.
Right.
And yes, and that experiment, the Codon plus one, that directly, clearly showed this thing.
Right.
Yeah, it's a wonderful result, sir.
Codon
Yes.
plus was the most stops all speculation. But
Right.
I also like the experiment which we took the silent mutation protein and took the protein and denatured it and it go back to the native omega.
Right. So like we face so much trouble expressing other proteins in E. coli. So maybe so we think that codon bias thing is more important. So just by introducing the tRNA will solve the problem. But the timing is also important. So the how much amount of that is present. So that is also important.
Very much. And not only that, Kamakshi, the whole point is now it is a very important field, this silent mutation. And you think of it, I don't want to pinpoint anything or make you biased. I may be thinking too much, but the whole point is when you look at the infection, including the present situation, virus mutation, which is happening all the time, different mutation which is killing us, what will happen to the silent mutations?
Right.
Right, the infective protein you are synthesizing, which is not generating antibody reaction in the host, but is a silent mutation, is a different structure. So, it's a very important area, and silent mutation will be killing for all of us.
Yes. So people are not like pointing on those mutations. We are all bothered about the mutation that is giving in the amino acid change. Yes. So yeah. So I guess questions have started to come. So from Iftikhar Rahman, sir, thank you for your wonderful talk. I have one question.
Sure.
Binding efficiency of all the sigma, that is sigma 32, sigma 54, sigma 70, etc., with RNA polymerase affects similarly in all the omega mutants?
Yes, very much, very much. Very good question, Iftikhar. You have done a wonderful question to me because I was bothered about this. Because when you look at the differential gene expressions, we find several genes are affected differentially. But I could not distinguish, I could not classify them that sigma 32 genes are altered better than sigma 70, than sigma 7, stationary phase sigma, etc., heat shock sigma. So I could not group them. But I'm sure there will be some subtle differences. So you have to pinpoint them. So I could not do that. But it's a very nice question.
Also, like for the in vitro transcription assays and all, so should people include omega in that too? Like, what do you think about?
You have to do that. In fact, later internationally, people who do crystallography of RNA polymerase, Murakami, Shettas, all of them told me that omega is an essential requirement for crystallization. Without omega, RNA polymerase will not crystallize. So the protein folding order depends.
Right. So that must be affecting the functionality that we see during the in vitro assays. Absolutely. And like all the experimentalists, they derive conclusion without the omega. So they should include, right? We all should include it.
Absolutely.
Yeah. So, okay, let's see. So yeah, Iftikhar is thanking you for. So. Let's wait if more questions are coming and we can address them. That's a really
can I ask a question, Kamakshi?
Yes, sure. So
this is Ruxana Chaudhary.
Ruxana of IICB?
Yes, the Dipankar.
So nice to see you. So nice to see you. Not see you.
And it was, hear me. It was such a pleasure listening to you after I'm hearing you after a long time.
Yeah, but I was just talking about you. Somebody, one of your students, I think, has joined us.
Yes,
Amit Vaidya.
Yes.
Amit has joined and he was telling, in fact, he is taking my lab. See, I'm closing my lab. I'm an old man now. So Amit
No, yeah. No, I can't agree with that.
So Amit told me that he worked with you.
Yes,
So I was inquiring
yes,
about it.
yes.
But I'm so unfortunate that I can't see you in person. Okay, tell me.
No, firstly, I must say it was such a pleasure listening to you. Clear, step by step for the students. What an excellent teacher you are, Kamakshi. I'm sure you'll agree. Excellent teacher you are.
Yes, definitely, sir.
Step by step, clear presentation for the students. Lot to learn from you, Dipankar. But my question actually is also about the ubiquitous presence of the omega factor in because if this is a chaperone, usually what we find is that chaperones, after the protein has folded into its mature form, has got to be dissociated for the protein to be functional at the cost of ATP maybe for protein.
Right.
So does this also leave the RNA polymerase at different stages of the translation steps, that is open complex, ternary complex?
Yeah, it leaves is very loosely bound. So as it processes open complex, closed complex, these are forming. Omega is dissociating, associating continuously. In fact, it has been shown by Richard Imbright. I recently heard the talk. I missed the paper. He published in Science, I think, recently. A transcription group meeting because of the COVID, Wisconsin meeting is stopped now. So they have an evening transcription meeting. So last week, Richard Imbright was talking. He was talking about ribosome movement on a coupled transcription translation. He showed that under certain condition, when the coupled transcription is going on, first ribosome, when sitting over RNA, and RNA polymerase is bound to it with a linker, it squeezes out omega for the facilitating the movement. So omega has several functions. Coupled transcription translation, omega is a function.
And I was just wondering about the silent mutations. Do you think they would have that kind of an effect in eukaryotic systems where?
Very good question. I don't want to burn my finger by commenting on this because silent mutation, first of all, I think it will have a bigger ramification, much bigger ramification in virus effect, eukaryotic. Not much work has been done. It's very, very unclear field, but geneticists should take it up. They should work on this.
Oh, yes. Okay, that's it. Thank you so much once again.
Thank you. So nice to talk with you.
Thank you, Rukshanadi. So moving on to next question from Shwandip Pal. Thank you for this exciting talk. So whether codon context is also playing a role in protein folding?
Codon context will definitely play a role in protein folding because protein folding, as I showed you, depends upon the rate of movement of the ribosome. So if the ribosome movement is altered as a change of the codon, then the protein obviously will have more time or less time as a folding, and therefore they will have an effect on the folding. That's the whole point. Minute effect maybe, but finally all proteins should go back to the most stable thermodynamic structure, minimum free energy conformation. But in the process, it will take certain, that is a bungee jumping kind of, it's a folding well. You have seen all those beautiful pictures. As it folding, there may be several times they'll spend in different positions because of the codon, of course, codon usage.
Okay, so moving on to next one from Vidhi Agarwal. Thank you, sir, for this illuminating talk. Can a group of silent mutations bring about a greater impact in the organism?
Yes, I believe so. I believe it will have but I am very worried these days after COVID that which mutations are good, which are bad, we do not know. Gain of function, the silent mutation gives a gain of function mutation, which is good for the organism. Then we are finished. It is bad for the host. So we should be able to, but whole point is we cannot treat a silent mutant. We do not know what is a mutation, right?
Right, it's so difficult. So next question from Shujay Chattopadhyay. Thank you so much for such an insightful presentation. While the role of silent mutation is badly overlooked, just out of curiosity, would like to know if you have checked how frequently that silent mutation is naturally occurring, as could be detected from the database, or any other such silent mutations.
You can look at the database. It is there, very much is there. Because in the silent mutations now getting popularity because of ITPs, internally disordered proteins. But you can look at the database. A database is being building up also for the silent mutations.
Yeah.
You do Google, you will get all the
So there was a second part of his question. I just missed it. Sorry to interrupt you. So basically to know if anything is known about the evolutionary convergence of the silent mutation like many others.
No, I won't be able to answer that. I don't think much is known, but I'm sure it has a role. Evolutionary convergence is a good question. It definitely has a role, but I don't want to give you a case on that because I have no clue on that answer. Okay.
Yes. So I don't see any more questions coming. So in your opinion, so like if we want to concentrate on the silent mutations, so we should initially look for those which have change in the codon usage or codon. Right.
Best is to look at simple IDPs, internally disordered proteins, and ask the question, why they are disordered, what is the function? If I make it ordered, changing a mutation, why silent mutation, would it have the same effect?
Exactly. So yeah, if no other questions are coming, then I believe we have to end the session here. So again, thanks a lot.
It's my pleasure. It's such a pleasure to give in talk, in particular in West Bengal, because biology still attracts people.
Definitely, sir. Such a such a topic you chose, and thank you so much from on the behalf of.
And I'll definitely come to your place someday.
Thank you. That will be wonderful. So we are so looking forward to have you at our institute. And on behalf of CHES, GIS, ISR family, I convey my sincere gratitude for your time. Thank you so much, sir.
Thank you, Kamakshi. Thank you very much. All the best. Shall I switch on?
Yes, sir. I think we can end the meeting now.
Thank you very much.
Thank you.